Somebody check, make sure I'm live. Maybe it's a delay. We'll go ahead and start. Hopefully it's recording, and then you can let me know. Send me an email. Okay, I've got 47 of you in here. Do any more want to get in?
You're live.
Okay, Swedish. Is that the right angle? Can you see the board? Do I need... All right. Make sure I keep going. Okay, are we in? All right. So, we're ready? Okay. So, chapter 9, cell cycle mitosis. Take a look at the following and see if these make sense to you. I'm going to read them because probably they're hard to see on the screen. Let me see tell me if this is better. Corbin, you're looking at it, right? Is that any better or worse?
My Wi-Fi stopped.
Oh, okay. That's a little better. A little better? Okay. So I'm going to read them real quick out loud. Can you describe... Hi. Can you describe the events that occur during the cell cycle? Right? So that would be like G1, S, G2, M has mitosis and cytokinesis and what occurs in all of those. How is the cell cycle regulated? Can the cell cycle ever pause? Can it stop altogether? Pause and stop? How does mitosis ensure the transmission of chromosomes from one generation to the next? What role does mitosis play for an organism? What occurs during the sequential series of steps of mitosis? What are the two types of disruptions to the cell cycle? Can you think what those are right now? Uncontrolled growth, which would be? Or when you stop, or when the cell dies, what's that called? There you go. What are the two categories of genes that contribute to those disruptions? If we talk about cancer, what are the two types of genes? I don't like the angle of the camera and the way it's on me. Sorry, I'm just step over here. What are the two kinds of genes? Yeah, oncogenes. Well, oncogenes is when it goes bad. What are they called? Proto-Oncogenes and tumor suppressor genes. Good. And do you know your highly suggested reading and thinking? Okay. So those are the big ticket items. Hopefully you've highlighted what ones you think you need to work on. And I'll go through the review right now. First thing up is the cell cycle, right? So you want to just remember what happens. So during G1, the cell is growing, but we don't have to memorize that because we know what stage precedes and the synapse going around. What stage precedes G1? Mitosis, right? And cytokinesis. So the cell just divided. So it makes sense that you need to grow because you're half the size. That makes sense? And also you're going to need more organelles. So that's when that happens during G1. This is also when you can pause for a little bit. When you're like, no, I don't need this right now. You might pause and go into G0. You might go into G0 temporarily. You might go in there permanently. Okay? Following G1 is the S stage. What does the S stand for? Synthesis of what? DNA. This is when you make the what? Yeah, it duplicates. What do we call them? Sister chromatids held together by a centromere. And ultimately, they're going to get separated during Anaphase. Good. So you're making the sisters. Do they appear as rod-shaped chromosomes at this point? No, it looks like something you pulled out of your hairbrush. It's just a mess. But the DNA has replicated at that point. Okay, G2, what are we doing? Proteins. Are we going to need a lot of proteins? Yes, for the next stages that we do, right? And then mitosis, remember, we want to focus on mitosis is what kind of division? Nuclear division. It's all about the nucleus. When you talk about mitosis, you're just talking about nuclear division. You're talking about the chromosomes becoming visible. You're talking about them lining up and separating so that each daughter cell gets the total complement of chromosomes. Because remember, mitosis is for growth, making more cells, repair. There we go. Sorry, I keep losing you. It just so happens that the last stage of mitosis, which is what? Telophase, happens at the same time as cytokinesis. Okay, questions or concerns on this part? Yes?
Do we have to know protophase?
No. No, you do not. Just prophase, metaphase, anaphase, telophase, cytokinesis. Yes?
Can cytokinesis be a part of mitosis?
No. Mitosis is division of the what? Nuclear material. Cytokinesis is division of everything else. Now, in the cell cycle, you're alternating between interphase, and they call this whole thing the M phase, but M phase consists of two things, mitosis and cytokinesis. So it's part of the M phase, but it is distinct, right? Distinct from mitosis. Yes?
Does D0 happen like during D1 or after?
During. And where's the sign-in paper right now? So it's going to go up and then down and up and then down, and then you guys will put it back. Okay, perfect. Can I keep going? All right, I'm going. So do this little review real quick. Drag your dots. So for those of you at home, the different stages, we'll go through them, and then you want to identify which stage does each of these happen at. Okay, here we go. All right, guys, help me out on this one. At the end of this stage, each chromosome consists of 2 attached chromatids. So when do you have 2 chromatids at the end of this stage? Which one is it? S stage. Good. During this stage, daughter chromosomes are distributed to 2 daughter nuclei. What would that be? M stage. That's what mitosis is all about, is distributing the chromosomes so that you have clones. Now, okay, hi, sorry again. We talked about this in relationship to traveling, and what was the ticket in our analogy? Cyclins were the ticket. And CDKs have to bind with, it's like getting worse. Sorry guys, hang on. There we go. Okay, we talked about you need the cyclins, and the cyclins will combine with what? CDKs, which are cyclin-dependent kinases, in order to trigger that next step. Okay, so the cyclins are the tickets, and these checkpoints are just like checkpoints in the airport. We need to make sure that the step is proceeding as it should. If it's not proceeding as it should, we need to stop and try to what? Fix it. If we can't fix it, then apoptosis. Which checkpoint is critical? G1, because we just finished mitosis, and did we screw it up? All right. Now, the signaling, because this is all underneath cell communication, so you get different signaling in order to make the cyclin that you need to get the next step to go forward, right? Because remember how we said the cyclins will come and go, but the kinases are always there. But those kinases might need to get phosphorylated. There's some other checks and balances to trigger to make sure that we're doing it the right thing at the right time. Yes?
Are those the only 3 checkpoints that are there, or are those?
The only 3 checkpoints are the 3 main checkpoints. They're the only ones you need to know. Okay. All right. Next. This is that cyclin CDK that I talked to you about. And then gradually you start to have cyclins build up. And when those cyclins, I put it all on one slide for you. When the cyclins build up, that'll trigger the CDK. And that cyclin-dependent kinase then will take a phosphate group, and it can take a protein that was off and turn it on. Or conversely, it can take a protein that was on and turn it off. And you have different cyclins getting elevated at different times. What is controlling what cyclins are getting elevated at different times? Where is that information coming from to build the cyclin? From the DNA, right? So it's what genes are turned on when, so that you get that protein built. Okay, conversely, it's because you can see it. Sweetheart, just listen to. Okay, then here we can see the chromosomes line up in the middle. What is the adaptation? Why is that so good to have all those chromosomes in the middle? Yeah, you can equally distribute them, right? You can make sure each daughter cell is going to get one. Each daughter cell is going to get one. The process where you separate the sister chromatids from each other, that pulling away like that, And that is all happening by these microtubules pushing and pulling and getting them all lined up and then yanking them apart. That is called anaphase. That's why we remember away on anaphase. And then at telophase, you're repackaging and reforming a nucleus. So you're forming the nuclear envelope. The nucleolus will start to reform, and then the chromosomes will start to decondense. They will start to unravel, so to speak, back into where it doesn't look like a rod shape at all. And simultaneously, you're getting this cleavage furrow here because it's an animal cell. You're getting that pinching off. Whereas in a plant cell, what you end up getting is a cell plate. And I didn't show you this in class, but this would be a really good review for you. I'm going to show it to you right now. See, do you recognize any organelles? What's that? Mitochondria. Sorry. What's that? Golgi. What's this? Nucleus. Nucleolus, good. What are those? Centrioles, good. What are these coming out? Microtubules. So they meet in the middle at metaphase. Away at anaphase, to nuclei at telophase. So you can see the two nuclei now. And now what's happening? Cytokinesis. Okay. So you have a link to that. There are like literally thousands of videos on that. Okay, so let's go through the song real quick on that. Here we go. The whole thing's called
Cell cycle.
mitosis. Let's go again. The whole thing's called mitosis. The first stage is prophase. The chromosomes become visible. The nuclear envelope and the nucleolus disappear, radiating microtubules. Asters, they meet in the middle at metaphase, away at anaphase, to nuclei at telophase, cytokinesis. That's it for an animal cell. We just did a cleavage furrow, or we would build a cell plate that will become a cell wall. Cell plate, plate, then wall, plate, then wall. Yes?
So when it grows the microtubules,
Yes.
is that the asters or is that?
The asters is that area where they're spreading out like everywhere.
So the area is called asters, but the part where
This right here would be an aster, but all of these are microtubules. See how it kind of looks like an asteroid moving through space? That's how you would think of that. Okay, any other questions on this? I'm looking. Okay, you're giving me thumbs up. Good. You can contrast this to what prokaryotic cells do. Prokaryotic cells, where's the sign-in sheet, by the way? Oh, sweet. Prokaryotic cells do not do mitosis. Do they have a nucleus? No, they don't. They have a single circular chromosome that replicates. It'll attach itself on either side, and then you'll rebuild a cell wall, and that's how you have your two cell walls. Can you see the plasmid in there? Yes. Do you see how the plasmid replicated itself? Okay, good. And what is a plasmid? Extra ring of DNA. Good. Then this is in here just for you to review, okay, about the functions and the different ones. Okay? Now, then this brings us to when things are out of control, and this is your cancer part. And so we talked about normal cells. These are two big things normal cells do. They stop dividing when they're crowded, okay? That's density-dependent inhibition. We're crowded, I'm not going to make any more. That's what normal cells do. And the second thing they do is they have anchorage dependence. They have to be attached to some other cell or substratum in order to survive. They don't have those two things, they automatically die. Okay, whereas cancer cells, they don't have that. They don't have contact inhibition. They just keep going even though they're crowded, and they don't have anchorage dependence. Now, was there an exception to anchorage dependence we talked about? Any in normal cells? I think I heard it. Yeah, blood cells, red blood cells, white blood cells. Okay? So we talked about a lot of things about them. We talked about that, you can go back in your notes how their nuclei are different. It's very coarse. They can have multiple nuclei, very little cytoplasm. They're unspecialized. We talked about that. They don't obey the cell cycle. And what takes them down that pathway? Well, we have two things. We have Proto-Oncogenes and tumor suppressor genes. So both of these have to fail in order for you to get cancer. Not just one, both of them have to fail. Okay, 2 fails. So proto-oncogenes are genes that are in a position of what? Power. And because they're in a position of power and they control so much, which is they promote the cell cycle. And how many different ones of them are there? Like 100, right? And if they go bad, then that causes a lot of problems because they're saying go cell cycle, go. Right? If those genes are coding for proteins they shouldn't. They're coding for cyclins that they shouldn't, let's say. Okay? But what keeps those bad generals in check are tumor suppressor genes, because these are like assassin genes whose job it is to pause a cell that's out of control, try and fix it. If it can't fix it, it what? Kills it. So both these things have to go wrong in order for you to get cancer. Okay? Proto-oncogenes have to mutate into what? What do we call them? Oncogenes. And they're saying, let's go. And I gave you an example of that. RAS. RAS is like stepping on the gas. The signal is not there. The receptor is not getting triggered by a signal, but yet RAS. So there's not a problem with the signal or the receptor, but in this case, there's a problem with RAS. It's acting like it's getting triggered when it's not. It's being told the light is green when it's not. And so it's telling the cell, hey, transcribe that DNA, make this protein. Do this transcription and translation. Okay, and then I gave you another couple examples where those breast cancer genes, right? That doesn't mean there's other ways to get breast cancer besides this. These are just 2 genes that promote that and facilitate it. It's all about go when it shouldn't. And then on the other side, your tumor suppressor genes, when you have those out of control cells, they're to push the pause button. But if you can't make the brake pads, you can't stop the car. That's what their job is to do. So we talked about the tumor suppressor gene, specifically P53. And if you have a functional P53 gene and it is triggered through signal transduction, if it is triggered, it will then make the breaks that you need for that cell cycle. Not only will it make the breaks, it'll do the killing through caspases if required. But if your P53 gene is not coding for the right protein, if it's in the, remember how we always talk about proteins being super specific, right? And so if it cannot have the right shape, then it can't do its job, right? Structure equals function. So it loses its functionality, and if it loses its functionality, then you can't stop the cell cycle. So it's allowed to throw its tantrum and be out of control because nobody's putting it in timeout. Okay? We talked about retinoblastoma in that too, the RB gene, but that was just a side note. Okay, my friends, that is it. Do you have questions? Thank you, Mrs. Sloan. You're welcome. Okay, bye. I'm sorry that recording was, hopefully that worked for you.